AI and organoids uncover early human development. Studying early human embryo development is vital for improving reproductive and regenerative medicine. However, ethical and technical barriers limit direct research on human embryos. Instead, scientists use models made from human embryonic stem cells, HESCs, grown in 2D or 3D to study development, disease, and drugs. A team led by Professor Jianping Fu, University of Michigan, created a special model using microfluidic and stem cell tech. It mimics key events that happen in the first 21 days after an embryo implants in the uterus, like the formation of the amniotic sac and the start of gastrulation, when cells begin to form different layers. A major challenge is that these lab grown organoids often develop unevenly and can look very different from each other, making it hard to study specific stages. Limited data and the need for manual analysis further complicate research. To solve this, a collaborative team published a study in Science Advances. They developed a better 3D suspension method to grow more organoids more consistently. They collected a large dataset of fluorescence images at multiple time points, showing how the organoids changed shape and which genes were turned on. They used AI to analyze these images. A deep neural network processed and segmented over 3,000 images from organoids, started with different cell counts, and grown for 12 to 64 hours. The AI created precise outlines of the entire organoid, the amniotic sac, and even individual cells. This allowed the team to pinpoint crucial milestones. Around 40 hours, GATA3 plus cells, a specific type, first appear, mixed randomly with other cells. After 40 hours, these GATA3 plus cells and T plus cells begin to sort themselves into separate groups on opposite sides of the structure, a process called symmetry breaking. By 60 hours, the GATA3 plus cells flatten out, forming a distinct asymmetric shape that closely resembles a real human embryo. The AI didn't just analyze the images, it learned the hidden patterns within the data. It used a model to predict the continuous developmental journey of the embryo from just a few discrete time points, filtering out random noise. This suggests that even complex biological processes might follow fundamental statistical rules, a promising idea for future research. First, the AI didn't just analyze the images, it learned the hidden patterns within the data. It used a model to predict the continuous developmental journey of the embryo from just a few discrete time points, filtering out random noise. This suggests that even complex biological processes might follow fundamental statistical rules, a promising idea for future research. Second, key findings, cell movement and AI predictions of research identified two critical time points, at 40H and 50H, where major changes occur. The most important finding was observing how cells self-organize to break symmetry and form distinct layers, mimicking natural development. By using AI to analyze the hidden features in the images, the researchers could predict how the organoids would develop over time. This ability to forecast continuous development from snapshots is a significant advance, hinting that complex biological systems may be governed by predictable patterns.